joining Science Talk Show presented by Times of Biotech. Today we have with us Professor Francisco Enguita from Lisbon University. He's well known for his research in non-coding RNAs, specifically those involving human diseases. Without any further ado, let's welcome Professor Francisco. Hello, sir. Welcome to the show.
Thank you very much for having me, and good afternoon for you, okay, and good morning.
Sir, to begin with, I would like to ask, what inspired you to be in the research career and specifically to research on non-coding on RNAs and specifically with the human diseases?
That's a very good question, but it has a very long answer. Okay, so the thing is, I study pharmacy. Okay, so because of family tradition, I'm the fourth generation of pharmacists in my family, and yeah, it's a very long story. Yes, and I went to the faculty, and I was not very enthusiastic about pharmacy. You know, I wanted to be astronomer. I wanted to be physicist. I want to look at the stars, but I realized that my skills in maths and physics were not enough. So I engaged this course, and very quickly I realized that the pharmacy was a very wide discipline, because we study everything. So we study chemistry, we study physics, we study biology. So, and very quickly I realized that I was able to talk with many different people. So I was able to talk with chemists, with medical doctors, with biologists, and I wanted to engage research because of that. So I engaged a small lab, a microbiology lab in the faculty, and I was working during my degree in the afternoons. Whenever I finished the classes, I went to the lab and did some basic research in microbiology. So I got a master's degree with this work, and I realized that I wanted to learn more, and I moved to the molecular biology field. And at the time in Spain, so I did my degree in Granada, in the south of Spain. So and at the time, we are talking about the mid-90s, okay? So at the time in Spain, there were just a few labs that were doing molecular biology, okay? And I engaged a lab that was in the north of Spain, and they were working with antibiotic biosynthesis, okay? So, and I did a PhD in molecular biology, studying the biosynthesis of beta-lactams, okay? These penicillin-like antibiotics. And during the PhD, I realized that I wanted to learn more about structure and function of molecules, okay? So I was very lucky, because I had the opportunity to apply for a fellowship here in Lisbon. And I got a very prestigious fellowship from the European Molecular Biology Organization, from the EMBO, to do a postdoc in X-ray crystallography. So I was working as crystallographer for 8 years. Actually, I have to confess, it is my passion, and it will be always my passion, the structure of the molecules. But at the time, I had no possibilities to continue because of no opportunities. So I engaged this place, which is the Institute of Molecular Medicine in the Faculty of Medicine. I started also to teach in the faculty. So if you ask me what I'm doing for a living, I will always answer you that I'm a teacher, okay? So I'm a teacher that in the free time I do some science, but my main job is teaching, and the only thing that I can do in my life is to teach. You know, it's the only thing. So
you are all passionate about teaching.
Yes, it is very important. I think it is very important, and you never realize the power that the teachers have. Okay. We have a very strong power, because we can change the mind of the young people, and that's amazing.
Yeah, in fact, the teachers build the nation, because whoever you are, first you have learned from your teacher the main concepts of what you are going to be. So of course, teachers have a really good, like they have a nation in their hand.
Exactly. That's the point. So and people people don't respect us, okay? But we have a lot of power because we can modify the minds of young people, and that's very, very important.
I know.
So talking about the non-coding RNAs, I told you that it was a long story.
Yeah,
But I mean, I got the position here to teach, and I was involved in some projects about a very rare diseases, and we realized that a specific disease was caused by a mutation that involves the extension of a gene. So in the patients, you got a longer gene than in the healthy people, and we tried to study this following the idea that the protein was changing, okay? But in fact, the thing that was important was the RNA, okay? It was not the protein. So I started to look at the RNA by many means, and I came out to the field of the microRNAs. So it was 15 years ago. At the time, nobody here knows anything about microRNAs. So I study and I started this field here. And now I'm more involved in other RNAs, like the long non-coding RNAs and the circular RNAs, which are a very new topic. So, and I'm trying to apply this knowledge to human diseases. I'm working mainly with cardiovascular diseases because we have the hospital here, you know, and it is very easy for us to get in touch with the medical doctors and to have this kind of, you know, dialogue with them. And it is always nice because they have the patience and we have the knowledge. So that's
it's a combination.
Nice. Exactly. It's like that.
Okay.
Yeah.
So I think while doing this combination, I think you have also published a very interesting concept. I would like to tell that it's interspecies communication in Holobiont by non-coding RNA exchange. So, sir, can you just briefly explain what is this concept?
Yeah, this is very crazy. You know, I mean, the thing is, besides the work with the medical doctors, I like to explore this kind of new concepts. And you know, you read the title of the paper, and the title of the paper has a word which is very strange, which is Holobiont.
Yeah.
So what is a holobiont? Basically, we are holobionts. Okay. So meaning that we are composed by cells that belongs to different species. So we have human cells, but we have bacterial cells, we have viruses, we have a lot of things. So all the organisms, all the big organisms are holobionts. So they are composed by many species that interact. And the idea of the study, how these cells communicate each other is very, very, I mean, very exciting to me. And I came out with this idea, putting together many knowledge that was published before. The idea that this communication between the different organisms that compose an holobiont may be due to the existence of an interchange of RNA molecules. Okay. The idea is that the RNA is a very old language, you know. So it's a very ancient language. It's probably one of the first biological languages that was invented. Yeah. So it means that all the cells are able to understand this language. It's like you know, like the Morse code. You know, everybody can understand Morse code. It's very old-fashioned. You know, it's very old, very classic. But everybody can understand that.
Yes.
So this is the idea. So the idea is that in fact we have cells that are communicating each other by using RNA molecules. Okay. And you have one example. Okay, I can tell you one example. Of course, this is not a biological, I mean, something that happens in healthy organisms. It is an infection. Okay, it's an infection. But this is an extreme example, but it is a good example for you to realize how it works. And the example is the hepatitis C virus. Okay. Yeah. So the hepatitis C virus is a very nasty virus which produces hepatitis C, and the hepatitis C is an infection of the liver. Okay. And for a long time, scientists were trying to investigate why this virus has this tropism. So the virus, why this virus infects the hepatocytes, the cells in the liver? Why? And they came out with the answer of this, because the virus is addicted to one small RNA that is produced in the liver.
Oh,
okay. Yes. So in fact, our hepatocytes produced a microRNA, which is a very short non-coding RNA, that it is specifically produced in the liver. Okay. And the virus uses this RNA to start the replication cycle. Okay.
Okay.
So this RNA, this small RNA, binds to the genome of the virus and somehow trigger the replicative cycle. So the virus is addicted to this RNA. So there is a communication between the host and the pathogen by this microRNA, this small RNA. And you can find many different examples in the papers and in the work of many groups showing that in many different cases. Okay.
Oh, it's quite interesting, and you nicely explained with the examples. Thank you so much, sir. But I think you have been working in interesting things, and the interesting concepts are not at over. So I want to go to another interesting concept of non-random genome editing and natural cellular engineering in cognitive-based evolution. So what is this all about?
That is very crazy as well. So this is actually is not my idea. So I was this idea was from a medical doctor called William Miller, and this doctor, he's really an amazing person. And I was contacted by him because I cited some of his publications, and he was very interested in my work. And he invited me to write a paper together with other scientists. And we came out with this paper talking about something which is very new and very controversial, which is the cognition-based evolution. And I mean, this is something that is a new concept, and it is something additional to the classical theory of evolution that starts with Darwin theories, okay? So you know perfectly the Darwin theories, and you know that after Darwin, when we discover the DNA, there is something called the post-Darwinian theories.
Yeah.
Exactly, where you put into the evolution, you put the DNA, okay? So you introduce the concept of the DNA as a major driver of the evolution. So, and this kind of concept, the cognition-based evolution, is different. So it means that the cells are intelligent. I mean, just consider that. Cognition does not mean that the cells have cognitive powers like humans have. Cognition came from the Latin cognoscere, meaning that you know, okay? You are aware of, okay? And this means that the cells are aware of the environment, exactly. So the cells, even if you think about a bacteria, so bacteria are aware of the environment. They are able to sense if there are nutrients in specific places, and they can move towards these places, okay? So they have sensors to somehow know what is going around them. So, and the cognition theory, the cognition-based evolution relies on this concept, relies on the concept that the cells are able to analyze the environment and are able to take decisions because of this environment. And the DNA is just a tool for these decisions. It is not the center of the problem, okay? So the cells have many tools to analyze the environment. And one of the tools that they use to take decisions is the DNA, okay? And of course, they can modify the DNA to be adaptive to this specific environment. And this is the concept. It is not very well accepted, believe me. Yeah?
Okay, so it's just a kind of a hypothesis which people are working on to make it get best out of this hypothesis.
Yeah, exactly. And if you can put together with all the hypotheses, you can put the non-coding RNAs as well as a signaling molecules. So you can put all together. So biology is more complex than we think.
Yeah, so this
sometimes.
non-coding RNAs can work as a signaling molecules, and with these signals and changing with the editing, we can we do something like make the environment or like the help them work against the environment, something like that?
Of course, of course, of course. All the time the cells are doing that. So they are adapting themselves to the environment. Yes, of course. Yeah.
So will it be like a genome editing tool for us also, like, because human is very much interested in editing DNA?
I know. This is, I mean, but of course the time frame is different, because when you are talking about genome editing, you are talking in a very short time frame for humans. I mean, if you apply these new techniques, we are editing just right now. Yeah. And when we talk about the genetic evolution by editing, it takes time. Okay, so the time frame is totally different. Okay, evolution is slow. Okay, by definition.
Okay, sir. So since I know that you are more interested in RNA, so I would like to ask you one more thing. That can we use this non-coding RNAs for these genetic disease treatments, or are we moving to that era?
Yes, of course we are moving. I'm very fast, believe me. Actually, the RNA field became more and more fashion due to the pandemics. Because there are two main vaccines that are made by RNA. So the RNA field is becoming very, very, very fashion. And of course, the use of non-coding RNAs for therapeutics and for diagnosis is very, very extended. Okay, and I will come back again to the example of the hepatitis C virus. Okay, so if you remember, so we have a microRNA in the liver that is used by the virus to replicate itself. Okay, so we can use this microRNA as a target for stopping the spreading of the virus, and in fact, it is used. Okay, so there is a drug that is coming into the market. It is now in clinical trials that use exactly a complementary sequence for this microRNA to block the microRNA and to avoid the virus to take profit of this microRNA. And we can think also about other diseases where we can use microRNAs or other non-coding RNAs as a targets for curing the disease. And there are many companies trying to explore that, and many drugs in clinical trials, believe me. So we are moving very fast towards this field. Yes.
Yeah, so because I asked, because the genetic diseases are the most untreated ones, because we don't have any treatment. So
yes, there is a problem. There is a problem with the genetic diseases. Whenever you have a monogenic disease where just one gene is involved, this will be easy to tackle, for instance, using gene editing, for instance. So if you can imagine a disease that has a mutation, that it's caused by a mutation in a gene, you can tackle and you can correct this mutation by gene editing, at least in the cells that are affected. Okay, there are some efforts now to cure genetic diseases that are related with the blood, for instance, like the thalassemias. Okay, and there are trials using gene editing. Okay, for correcting these mutations, and at least you can correct the mutations in blood, and you will actually cure the more severe symptoms. Of course, if you are talking about other diseases that have widespread symptoms, you cannot tackle all the organs in the body with gene editing. Okay, so you just can tackle a few, but sure that we are moving to this in this direction. But if you think about genetic diseases that are polygenic, so there are many genes involved, this will be more difficult. Okay, this will be more difficult because in many of these polygenic diseases, we even don't know how many genes are involved and which are the genes. Okay, so for instance, we know that some diseases has a genetic background, has a genetic transmission, but we don't know how many genes are involved, and
actually involved in the disease.
Yeah,
okay.
That will takes time. Okay.
Okay. Yes, since you are a teacher also and you have been researching, I would like to put some in this hypothetical situation. Like sometimes being a researcher, we experience that we are repeating the experiments again and again, and we are not getting any results. And it takes months together, which was actually supposed to finish within a week. So in this scenario, what are the ways one can motivate themselves? And have you faced any challenge like that, or it is a hypothetical situation for you?
All the time. I mean, science is like that. I don't remember who was the guy that told this sentence that science is 5% of inspiration and 95% of transpiration. I don't remember. I think that it was a very important guy. I think that Newton or Einstein or whatever. So, but this is like that. So science is like 95% of transpiration. Okay. So, and I will always recommend the students that are engaged in a project that they apply the scientific method. Okay. So meaning that they have to listen to the data that they obtain, but listen very carefully to the data. Okay. And not trying to put any bias into the data.
Yeah.
So meaning that they don't need to introduce any preconcepts, any bias over the data. Sometimes you are expecting some result, you know what I mean? So you are expecting that things are going to give you this information, and sometimes things will not give you this information simply because you are wrong and you are putting bias in your experiment. So just listen the data, listen carefully the data, try to analyze the data by many means. If you are looking at the data by using software or by using analytical techniques, try to use another one, okay, to extract more information. And it's like an orchestra playing a symphony, you know. So you have the whole orchestra playing the symphony, and you have the music, but you can pinpoint all the instruments individually, and you can listen to the music that is producing any of the instruments in the orchestra, and that's science. So you have to listen to the data and never put any bias. Never think that that your results are going to be these ones or the other ones. So just listen. Don't put any bias. And of course, this is hard. Science is hard because especially when you are dealing with biological systems. I mean, biological systems are temperamental. So I mean, you know, sometimes the cells don't grow, they don't replicate. You never know what happens. So you have to be persistent, you have to be strong, and you have to be committed. And that's it. And listen to the data. That's it.
Yeah, we have to be ready that observed results can be much more different from than expected results.
Yeah, exactly. And sometimes this is the funny thing about, you are expecting some results and you get the opposite results. And sometimes this is the important thing. And and I mean, I never, for a student, never think that you are going to produce a very important
accurate one.
Exactly. Never think like that. So science is like a beach. Okay. So every scientist will put a grain of sand in the beach. Of course, a grain of sand is little, it's very little. But of course, many grains of sand, they will produce a beach, okay?
Beach, yeah.
So, but I mean, you never think that you are going to produce a very astonishing result. So you have to be humble, modest, and think like that. So the more, I mean, it happens to me. So the more I study, the less I know. So it's like that. So, yeah.
So in this on this beach of science, so you have you have put many grains of sand, and one of that grain of sand is like your miRNAtool web page. So can you give an overview of that tool?
Yeah, I like very much this page, but I tell you, this was born as a need for organizing my bookmarks, you know.
Okay.
So I got a crazy collection of bookmarks, of tools, pages, things, which were like a mess in my browser. And I thought, maybe I can create a page with all the links organized by topics. So this web page, for the people that don't know, is called Mirna Tools. And it is a web page that it is basically designed to teach people. So it has a collection of tools that you can use for free in the web to analyze, especially microRNA function. And I also have in the page a lot of tutorials for trying to guide the people how to use the tools and how is the best information that you can get from these tools, okay? Because these methods are not written in any paper and they are not written in a book. So you have to learn by yourself. And I thought that it was interesting to put everything together and to help people. So people to use how to use different tools to, for instance, detect microRNAs, to predict the function, to look at other data, to see if these microRNAs are expressed in a specific tissue, and all these kind of things, but in a well-ordered way, I think. Okay. So I use this page to teach in courses. And we used to do practical lessons and practical sessions, and we use the page to teach, because you can follow the tutorials and you can learn a lot. Okay.
So other researchers who are not exactly working under you, but they can also make use of this page, because the tutorials and everything, it's a combination of everything. You don't have to go anywhere to use these tools.
Exactly. So you have the links, and you can go to the tools, and you have examples. And you can follow the examples, and you can teach yourself, you can train yourself. I have their experimental data as well. So you can use this experimental data to play, and then you can go to your more serious data, okay? And to analyze your problems.
Actually, he contributed a lot to this beach of the science. So I'm really glad to meet you. Thank you so much for accepting my invitation.
I'm very happy, really. I'm very welcome. I'm very glad to be here. And you are very brave to set up this initiative. So I think that it's very important. It is very important to have these kind of initiatives, because science matters, and we need to spread science by using this kind of media and this kind of ways. So congratulations, and thank you very much. It was a pleasure to be with you.
Thank you so much, sir.